Welcome to Botany class. Today's learning objective is cyanobacteria. Before learning cyanobacteria, let's see what are archaebacteria. Archaebacteria are primitive prokaryotic organisms which adapted to thrive in extreme environmental conditions. So, this archaebacteria adapted to thrive in extreme environmental conditions like high salinity, high temperature or low pH. So, it can tolerate, that is archaebacteria. And this archaebacteria cell wall which contains lipids like glycerol and isopropyl ethers. Glycerol and isopropyl ethers are present in the cell wall. Due to the presence of this lipid like glycerol and isopropyl ethers, the cell wall is resistant to antibiotics and lytic agents. If any lytic agents are attack this, it can tolerate because of the presence of this glycerol and isopropyl ethers. So, it can resist, the cell wall resist the antibiotics as well as the lytic agents. What are the examples of archaebacteria? Methanobacterium, halobacterium and thermoplasma. So, these archaebacteria are adapted to thrive in extreme environmental conditions. So, it can be tolerated in high salinity, high temperature or low pH. Example, Methanobacterium, halobacterium and thermoplasma. Now, let us move to cyanobacteria. What are cyanobacteria? Cyanobacteria are commonly called as blue-green algae or cyanophyceae. Cyanobacteria. How old are cyanobacteria? How old are cyanobacteria? Cyanobacteria, there is a deposition of the colonies of this cyanobacterian members. Cyanobacterian members, when you are looking the thallus, it can be filamentous or it is a colonized form or it is a thallus like organization, different forms. So, this colonies of cyanobacteria deposits, it binds with calcium carbonate and its deposition is known as stromatolites. Now, how old are cyanobacteria? Due to the deposition of this stromatolites, how stromatolites are formed? This cyanobacteria, colonies of cyanobacteria which binds with calcium carbonate and deposited and forms stromatolites. So, geologically, its geological time, it's said that 2.7 billion years ago. So, this cyanobacteria, what are the salient features of cyanobacteria? These cyanobacteria are blue-green alga or it is commonly called as cyanophyceae members, cyanophyceae. So, this cyanophyceae lives in freshwater and marine water. Most of the species are freshwater and marine water species are present. What are the marine species? Trichodesmium and Dermacarpa. There are so many species, but you are going to learn only two. Marine species of cyanobacteria are Trichodesmium and Dermacarpa. And in the Red Sea, the color of the red in the Red Sea, because of the presence of Trichodesmium erythroium, Trichodesmium erythroium. Due to the presence of this, the red color forms in Red Sea. And this all cyanobacterian members which adapted in the form of endophytic and phycobionts. What is endophytic and what is phycobiont? Endophytic which lives between the living plants. The phytic it lives in the plants, living between the plants. For example, there are nostoc, anabina, oscillatoria, this type of organisms, cyanobacteria, the blue-green alga, these are lives in other plants, living plants like coralloid roots of cycas. This nostoc and anabina which lives in the coralloid root of cycas, And there is a species Azolla. Azolla is a water fern. So it also makes symbiotic association, and this all fixes nitrogen. So they are examples of endophytic species. Nasta, Canabina, Oscillatoria, they are this endophytic species. And what is meant by phycobionts? Phycobionts have the association of lichen. What is lichen? There is association of algae and fungi. Is lichen. So algal partner, phycobionts. Phycobionts are the algal partner. Here the lichen is the association of algae and fungi. The algae, it photosynthetically it synthesizes its food and supply to fungi. Fungus which collects water and give. Both are mutually benefited. So here the algal partner is phycobiont is Gliocapsa cytonema. Examples of phycobionts in cyanophycean members are Gliocapsa and cytonema. Now let's see what are the salient features of cyanobacteria. There are particular salient features. What are they? These organisms are evolutionary record says that it's a primitive prokaryotic photosynthetic organism. Because of the presence of pigments, it synthesizes its own food. So it is a prokaryotic organism, and it forms in different thallus organization, unicellular colony, as well as it is filamentous. Let's see few examples. Unicellular organisms in cyanobacteria is Crucoccus. This is the picture of Crucoccus. Unicellular cyanobacteria is, or cyanophyceae or blue-green alga is Crucoccus. Then colonial forms. This is the example of colonial forms, Gliocapsa. This is Gliocapsa colonies. It's present as a colony. And filamentous, nostoc. This is the filamentous alga, blue-green alga, nostoc. Nostoc is filamentous. And when you look into this protoplasm, the cytoplasm, which is differentiated into two regions, the central part is called as centroplasm and the chromatoplasm. So chromatophores. So two regions, the protoplasm divides. And certain species of oscillatoria, it shows gliding movement. Gliding movement is very important in cyanophycean members. Cyanobacteria, it shows gliding movement. One word question, oscillatoria is one of the example of gliding movement. And there are many pigments. Pigments are very important characteristic feature of alga. So this is coming under blue-green alga. The pigments, what are the pigments present in this? Phycocyanin, phycoerythrin and myxoxanthin. Phycocyanin is present because it is this is a cyanophycean members, phycocyanin pigment, and phycoerythrin because red alga is also present there, because due to the presence of this red color due to Trichodesmium erythraeum. So it contains phycoerythrin and myxoxanthin, myxoxanthophil. These all pigments are present in this cyanobacteria. Now reproduction. Only vegetative method, sexual method of reproduction is absent in cyanobacteria. So common vegetative method of reproduction is echinus, Harmogonia formation, fission and endosperm formation. Echinus are the thick walls. It forms as a vegetative cell. Around this there is a thick wall. So echinus formation. Harmogonia, here in this Harmogonia, filamentous alga which produces a cell division. So it forms a Harmogonia, and fission or endosperm formation, endospore formation. It forms endospore. and when the conditions are favor, it develops into a new species. So reproduction only vegetative method by means of akinase, hormogonia, fission and endospores. And most of the species of this is used as biofertilizers. And here it produces certain filamentous alga in the terminal region or in the intercalary region. It produces a structure called heterocyst. So this heterocyst which fixes nitrogen. Heterocysts are used for nitrogen fixation, and there is a mucilage substance outside the thallus. Due to the presence of this mucilaginous substance, it's also called as myxophyce. So it's called myxophyce. Actually it is cyanophyce. Cyanophyce. Due to the presence of mucilaginous substances, it's again known as myxophyce. And most of the cyanobacteria which forms water bloom. What is this water bloom? This abundance of, due to the abundance of this species, it causes toxic substances which cannot allow the proper sunlight and aeration towards the water. And due to this, it produces a toxic substance. Oxygen depletion causes, so toxic substances are released. What are the examples of water bloom species? Microcystis and Anabina. The name of the species is Microcystis aeruginosa. Microcystis aeruginosa and Anabina flasaceae. Anabina flasaceae. They are very important for causing water bloom. They are producing toxic substance and causes water bloom. So salient feature of cyanobacteria is very important. 5 mark question. So first write about the thallus organization and write about the gliding movement. Then what are the pigments present, and the reserve food material is cyanophycean starch. Reserve food material. Then how reproduction taking place. Then why it is called myxophyce, and what are the examples of water bloom. Now 2 more species, let's see. Mycoplasma. What is meant by mycoplasma? This is the structure of mycoplasma. This mycoplasma varies from, it's a microorganism which varies from 0.1 to 0.5 micrometer. The size ranges from 0.1 to 0.5 micrometer. Mycoplasma. And this mycoplasma is a gram negative bacteria. It shows pleomorphic. So it's a mycoplasma is a pleomorphic. It lacks cell wall. Outside there is a cell membrane. This is the cell membrane, but it lacks cell wall. So it shows pleomorphic nature. And it is first observed by Nocard and co-workers. Nocard and co-workers observed in the year 1898 from pleural fluid, because this organism which causes disease in plants as well as animals. It causes cattle, there is a disease, bovine pleuropneumonia. It causes bovine pleuropneumonia in cattle. From this pleural fluid, Nocard and co-workers isolated this organism, mycoplasma. See the structure of this, it lacks cell wall and the outer membrane is cell membrane, and the DNA strand, the DNA which contains low guanine and low cytosine. Cytosine and guanine is low in its DNA strand, and it also possesses ribosome. So this is bovine pleuropneumonia is caused by Mycoplasma mycoides. Mycoplasma mycoides causes bovine pleuropneumonia in cattle. It also causes disease in plants also. There are certain diseases caused by Mycoplasma. One more species, Actinomyces. Actinomyces. These actinomyces are commonly called as ray fungi because of its mycelial structure. Actually, the mycel outside, it never produces the mycelium. Due to the structure of its mycelia, it's called as ray fungi, and it is also gram negative bacteria, actinomycetes or actinobacteria. It contains high guanine and cytosine. So, compared to this mycoplasma and actinomycetes DNA which possess high guanine and cytosine, but mycoplasma which possess low guanine and low cytosine. Then there are so many species which shows symbiotic relationship. One of the species is Frankia. Frankia which shows symbiotic association. You know very well what is meant by symbiotic association. There is a mutual relationship between two different organisms. So, this Frankia is actinomycete. Frankia which shows a symbiotic relationship with Alnus and Casuarina. Alnus and Casuarina, it makes symbiotic relationship. And actinomycete bovis, which produces, which lives in the jaw, and it causes lumpy jaw. It causes lumpy jaw. Actinomycetes bovis. And in the rainy season, during rainy season, there are Streptomyces species. After a long time, when the rain begins, the earthy odor appears because of the presence of actinomycete. One of the species is Streptomyces. Streptomyces causes earthy odor due to the presence of geosmin. Geosmin, one word question. Due to the presence of this geosmin in the soil, the Streptomyces species which produces the earthy odor at the time of rain. So, these are activities of actinomyces. Actinomyces are commonly called ray fungus. It is a gram positive bacteria and it contains high guanine and high cytosine, and it shows symbiotic relationship. One of the example is Frankia. It makes symbiotic relationship with Alnus and Casuarina, and Actinomyces bovis which causes lumpy jaw which causes. Streptomyces cause earthy odor. Earthy odor is caused by Streptomyces species. So, once again let us review what you learned today. Archaebacteria, first of all you learn about Archaebacteria, then about Cyanobacteria. What are the salient features of Cyanobacteria, and what is Mycoplasma and Actinomyces. Draw these diagrams in the class note and learn all these questions. Thank you.